Uh, can we get the SparkNotes version? I'm
Yeah,
sorry. My internet sucks. I'm in Louisiana, so
yeah, go to graduate school in a more northern state. You'll be better off.
they'll probably
Anyway,
agree with me more politically there. Anyways, who said that? Get out
yeah,
of here.
yeah. Anyway, so all I was saying was that that people are, okay, science is this human enterprise, and people contribute to it and shape it in the direction it goes. And Haeckel was one of those people who's very influential, and he shaped a lot of the modeling of how life arose with things like his primordial ooze hypothesis, and he shaped a lot of things about how we thought evolution worked. Um,
That's true.
and and a lot of those ideas got discarded,
Yeah.
you know, gradually during his lifetime, but then some of them still linger on.
Right. Because
Yeah,
I mean, we still teach ontogeny recapitulates phylogeny in the textbooks. Right?
we do. All my textbooks say no, that's false.
What?
But at the same time, you get people who,
I was told that we all believe that. So, oh,
yeah, but you still get people like, what's his name? Paul Davies, who's basically so strongly influenced by that he thinks he can argue that cancer is a reversion to an ancestral form.
nice.
Yeah, and that's pure recapitulation theory, and it's totally it's totally refused as a viable hypothesis by all credible biologists, and yet Paul Davies makes bucks by proposing this wacky idea, and everyone is telling him no. No, there is no ancestral organism still preserved inside each of our cells, waiting to break free of its bonds and leap forth and cause cancer. That's just nonsense. But
Mitochondria just, chomping at the bit to escape.
yeah, that's it. They're okay.
That's does he actually attempt to argue like any particular genes, or is he just kind of putting forth the whole model at once, just saying
He's putting
this?
forth a whole module,
That's it. Okay.
and he's literally cited recapitulation theory as a foundation for it.
Yikes.
Yikes is right. Yeah, and it doesn't matter what people say. He established his credentials as a physicist. He must be a smart guy, and everyone knows on the scale of sciences, physics is the smartest, so much smarter than biologists, and therefore he must be right. It's kind of infuriating for that reason as well.
Yeah, I you did a video which I thought was very interesting on how was it embryogenesis mirrors oncogenesis, which I thought was kind of interesting.
Yes, but it's not that we're reverting to
Right.
a
It's just
form.
that it's kind of paralleling in some senses.
Yes, but cancers should be thought of as a new organism. If you're gonna
Right.
take that perspective, it's not an ancient organism that's come back to life. It is a new organism that has resulted from selective mutation and deletion of genes to give it new properties. Um, and that's an interesting approach to it. It's a really bad organism. It's kind of lost a lot of regulatory controls, and that's one of
True.
the reasons the problem. But um, sure, yeah, that cancers are, looking at where the money is going in the funding agencies, I would say cancers are really interesting.
Well, I don't know. I think we should put some of that money into looking at protists. That's just my opinion. And
Sure.
basal animals.
Oh, so you're on the side of Paul Davies, who'd be arguing for some of that cancer money to go to studying single-celled organisms,
There
right?
are there are enough people on this planet as it is. We can take some of that. I, you know, we're always talking about like the strain that this large a population is putting on the environment. We don't really need all those people.
Sure. Yeah. No, you're echoing some fairly common arguments here that come from the basic science community that there's way too much emphasis in applied research, in biomedical research, and
Of course, I say
those of
this in
us who are,
jest.
yeah, and those
Uh,
of us who are doing these other things are just getting the scraps, the leftovers from that money. That's not fair and not right, especially since nowadays we should be so much more concerned about the environment and ecology that um, it was one of the things that affected me was I'm sitting there thinking of future work I want to do, and I've always been a lab guy working on model organisms, and just thinking about the importance of ecology and the environment and how things interact, and we just sort of miss so much when we study a single organism that I decided, yeah, I think I'll start working on spiders.
he said, "So long, zebrafish."
Yeah, it was an abrupt decision on my part, and I just kept this zebrafish around till he died a natural death. Um, so I didn't. They're lovely animals. I
Yeah.
didn't want to utilize them, so I just kept them going as long as I could. And in parallel, set up this other system because what I'm finding increasingly interesting is not just here's this animal, it's really cool. Let's
Yeah.
figure out how it works. But this animal that interacts with all these other creatures and organisms and environmental factors. And let's start looking at those pathways that affect it, see what we can figure out. Of course, I just made myself less fundable too, but that's okay.
How did you do that?
Well, nobody wants to pay money for that kind of thing anymore.
Oh,
This, yeah,
sad day.
uh, biomedical funding, you get to apply to NIH for those big budget grants. And if you're studying spiders, you get to apply to in-house grants and NSF for little tiny pots of money that might keep you going year by year. Uh, it's
Huh.
just the way it works.
Well, time to change that. Time to get rid of biomedical research.
Well, now I'm torn because I'm also getting older and I'm probably going to need medical research.
Dang. I guess, yeah. I don't know. I think about the.
Keep it, keep it around until I've achieved immortality, and then we can talk about getting rid of it.
We'll see, that's exactly why we need to. We'll see, we get rid of it. We go back to studying the Hydra, figure out how it lives forever, and then we go back, utilize its whatever genetic systems it's using. So, because I
I
read a paper on that. So,
oh, okay. Yeah, and I think though that immortality for us is only achievable if we strip ourselves down to just the bare minimum, like a Hydra. Um,
and then just try to go about it as a little Hydra.
yeah, let's turn ourselves into squidgy little things that live in the water, and um, it would probably be a good niche if we got rid of all those big mammals that are poisoning everything.
Yeah, that is probably the case.
Uh-huh.
Yeah, I was thinking about future uh, projects, stuff like that. A few weeks ago, and I was kind of thinking about how few like molecular systems we just have the evolution of them mapped out to a very high degree. You have things like a ATP synthase, where they have kind of the step by step stuff of this enzyme was, reappropriated and brought to this other thing, and they were kind of brought together. And there's not really a whole lot of examples. Like I guess the bacterial flagellum is another, where you have a bunch of different, the genes are homologous to each other, and you have these different proteins which had various jobs and all that. But there's not really a whole lot of that, and I guess it's probably because there's not immense funding in that area.
Yes. Well, but if you get into biochemistry, you'll find more examples of people trying to reconstruct the evolutionary history of single enzymes, for instance. And there are directions you can go in for that. And I'd also mention that there are things like when you're trying to puzzle out who the initial herbalitarian was, that's led to this discovery of receptor tyrosine kinases that are present in animals, but nothing else.
Hmm.
And you can use that as a marker, and then you can and of course the other thing is you find that gene product that's got all kinds of variants and expressed only in animals, but then you look outside the animalia and you find that in plants you've got receptors and you've got tyrosine kinases, and they're separate. And it's really easy to put together a model for how that, it's a fusion protein basically, that
Right.
enhanced the capabilities of the ancient animal that evolved it.
I think you did a lecture on was it Stephen Meyer's book, uh, Darwin's Dad, way back where because he makes the argument that for there's all this complexity, it just comes out of nowhere in the Cambrian radiation, and obviously there must have been more and more genes that just popped out of nowhere, but it's like as you were pointing out there, and it's like that's also work by King and Burnett and those other people,
Uh-huh.
um, that there are RTKs in choanoflagellates and Philisterians, Nyctiosporians, and like integrins and things like that.
Yeah. Yeah, that and when you think about it, when you dig into it, you discover that okay, crustaceans and people share a lot of the same genes. How do you explain that? The easiest
Magic.
way is, oh yeah, well, I mean the easiest way that uses evidence
Oh, okay, well,
is
fine. There's that,
is
I guess.
common ancestry. And yeah, that's exactly the position the intelligent design people are in, is they are arguing for magic. And
Yeah.
there's no way to demonstrate any kind of magic. Now, earlier we were talking about how a lot of these phylogenies that are assembled are incomplete, and they're hypotheses more than established fact. But still, there's something. They're working in the right direction, and they're giving us clues for what to do next.
Right.
And ideas like intelligent design, just they're dead ends. Um,
Right.
how am I supposed to test intelligent design unless I've got my intelligent designer trap?
You got your magic wand, and you say, "All right, students." Yeah.
Yeah, or last night I put out a trap and I caught God, and I'm bringing him into the lab, and we're going to dissect him today.
Lured him in with candy or something.
Yeah,
uh,
yeah. Into a blender and we're going to start extracting genomes.
uh, yeah. The oh goodness. Even like organs. Sometimes there's not really a huge amount of data on it. Like I was looking for stuff on the evolution of the kidney, and it's kind of surprisingly difficult to find.
Well, yeah, the details are complex, but the origin of the kidney seems to be pretty straightforward, because you recognize that it's a mesodermal structure that's in the
Mm-hmm.
coelom, and it's ciliated, and it's doing all these interesting things to move fluids around. Um, it gets all tangled up in the reproductive system early on,
Yeah.
because
The
reproductive system is doing something similar. And then there's this divergence into these really specialized functions. Yeah, but again, we can put together a lineage for that. That's a good hypothesis, and we can also find variations in extant organisms, and we can see all the different pieces that have been assembled for that. It isn't a unitary whole that appeared magically as one thing. It's pretty clearly, let's assemble it from these precursors that gradually become more complex.
so would you say you follow the was it the tripartite hypothesis on that?
Of the kidney?
Yeah.
You're always doing this. You're trying to pin me down.
Because
And
I literally found out about the was it holonephros versus the tripartite hypothesis just last night as I was
yeah,
reading about it.
Yeah, I mean, there's right now you look at the kidney and it's this beautifully organized structure with these specialized zones that often are built by the same cells traversing the zone, et cetera. And it seems like something very well integrated.
Hmm.
But you could imagine that there was a precursor that was not quite so tidy that had, different regions specialized for different functions.
Always trying to take the middle road, PZ.
I know I'm so wishy-washy
I
that
wish
way.
that we could just like assert things and then it were fact.
Yeah, that
Oh,
would
to
make
be a
life.
creationist. Oh well.
Yeah, yeah. There's one true path, and it's the Myers model of everything, and you have to follow it.
Oh, that's why I had you on. See. Yeah,
And then you discover that the Myers model is this goop that just kind of wishy-washy and says, "Okay, maybe this way."
yeah, maybe this way, maybe not. It is uh interesting that, or I guess, because I had to, I did a couple of videos about, or going back to Meyer, uh, I did a couple of videos about his calamitous book, and or uh, what do you call it, doorstop?
Yeah.
And one of the interesting things he says in it is that the Hox genes played no role, or couldn't have played a role in evolution and macroevolution. That's and so the interesting thing. Of course, he cites literally no examples. Uh, he has this.
Right.
It's only like a 3-page section on Hox genes in his book. He cites no examples, just kind of briefly mentions what they are, and then says all they do is turn things off and on, so they can't do anything.
Yeah, that's one of the things that drove my students crazy this past semester. Is I do a lot of stuff on gene regulation, and that's all. Here's how you turn genes off and on.
Right?
You generate patterns with the same sets of genes in this same field of cells.
Yep.
Um, and it was kind of alien to them because I think most of our students grew up on simple Mendelian genetics. That's how they think of genes everywhere, and
Yeah. I mean, I don't know
it's not
how
the
you
case.
guys
Uh,
teach genetics up there, but that was the genetics class I took was like Mendelian genetics on steroids.
yeah,
I actually, well, I probably shouldn't say it on air.
I'm teaching genetics next spring, so and that's one of my constant struggles in that class is that you've got to give them a foundation in simple Mendelian genetics, and they can understand the mathematics of that, and then you start bringing in epistasis and all these interactions between genes, and then you bring in the interactions between genes and the environment, and all of a sudden
Hmm.
you discover that no, nothing is Mendelian ultimately. That it's all gotten so complex and refined and variable that um, yeah, it's just not right to make Mendelian genetics your sole model for viewing
I mean,
inheritance.
we did talk about non-Mendelian inheritance, but that was more of the exception than the rule.
Right.
Things like um, what is it? Was it maternal imprinting or
Maternal effect,
maternal effect? Yeah,
printing,
yeah,
yeah, and epigenetics gets in there and all this other stuff. But you're right, it's usually taught as this weird little set of outliers. Everything is Mendelian except, there are these other things, and we just sort of throw them in there as an afterthought to say, it's actually more complicated than that because real geneticists will tell you that it's Mendelian genetics is not adequate to explain the phenomena we're seeing. Uh, but we kind of flip the priorities around.
hmm,
And I confess I do that myself because what you discover early on when you're teaching genetics is that most of the students are completely unprepared for the quantitative aspects.
sure, sure.
Yeah. And if you throw epistasis at them on the first day, nope, they will all flunk or withdraw or run away or something. So you give them
Gotta
that
warm them
familiar.
up to it, huh?
Yes. And then you discover, well, it's the second to the last week of the semester, and now I finally get to talk about the good stuff. But
Oh man, that's sad.
it is, yeah. But it's what we got to do. I mean, the whole idea with undergraduate genetics is you are not really teaching them the whole of genetics. You're teaching the basement foundation of genetics, and then
Hmm.
you hope, well, they'll be so excited about it, they'll go on to graduate school and they can learn the real stuff.
Right.
Yeah.
Yeah,
So,
I did some reading recently on I guess more of a historical paper, and I didn't. One thing I guess I didn't know is that we got pretty close to almost having Darwinian genetics instead of Mendelian genetics because Darwin basically came up with the ratios in primroses around the same time as Mendel with his pea plants.
well, he had some preliminary stuff that was going in the right direction, but the thing is that Darwin never applied that mathematical approach to it that
Right.
Mendel did, and that was key. That you could have, without the math, you did not have a method to dissect out the different processes going on. With the math, you can, for example, when you see a non-Mendelian ratio in in genetics, you have a tool to take it apart, interpret it when you use the math. Darwin had no way to discriminate between anything. It was just all as a commission of, well, here's how we pass things on to our next generation, and
Hmm.
how it works, I don't know.
Right. Right. Oh, this is very curious. I just realized there were two different chats. The first part of the video. That's so very weird. I'll have to sort that out later. Oof. It appears that like when we dropped, I think the video got cut in two. So so it's like it started
Okay,
streaming a
you got the
second
tools
video.
to splice it back up.
I don't think
Oh,
so. Well, it doesn't matter, but I think it basically had two videos. Yeah, going at once. Or we had the first one and that went, and then when the internet dropped, it split and started a second one, and so that the second one is live right now. And I just realized that because I was like, why is the chat not moving? Oh, that's why. Whoops. Well, I see everyone now. I see
oh,
lots of people posting things. My bad. Sorry about that, everybody. Yeah, I was still. You're right. It was really just kind of
okay.
preliminary
Well,
stuff. But um, I did not know that he was doing that work. Also, I was not aware of that, which I think is pretty neat because it just kind of or it increases the the respect that I have for the amount of research that he accomplished in his life. I mean, he found so many more fossils in South America of ancient mammals than because they only knew of two before he came on the Beagle, and then he found like 10 plus more. Went to the Galapagos, went to was it the islands in the islands where he did his coral research. I don't remember what they were. Australia, and then came back and did all the experiments and stuff, and just it blows your mind or blows my mind, anyways, how much he accomplished. So
yeah. And as an undergraduate, he was primarily a geologist and botanist, which is
yeah,
just, yeah. So you see, once you finish your undergraduate degree, you weren't done.
it's only just begun.
Yes. Who knows? You could end up doing something completely different in your graduate career, in your postdoctoral career. Who knows? You'll be working on.
Uh-oh, can you hear me? Oh, there you are.
I can hear you now. You dropped out for a while.
Oh, it was just a second. Yeah, that reminded me of what one thing I have a professor who said so many students become marine biologists to save the turtles, but then they get stuck studying a worm for the rest of their life.
Stuck. Worms are interesting. There's
I
a lot
mean,
of cool
worms are.
worms out
I
there.
think acorn worms are pretty interesting because they're on our own side of the line,
Yeah.
so that's pretty cool, you know.
Yeah, sure. And polychaetes, they're cool. Nematodes, they're cool. They're
I,
all worms.
yeah,
Yeah.
they're kind of cool, you know. They're they're in everything, in and on everything.
Uh-huh.
Um, what was it? I just watched a video earlier about the preapulids, the penis worms.
All right.
Yeah, they're an interesting group.
Uh,
Uh, I don't know. I think kind of rinks are kind of neat. You know, they just
yeah,
found like some of the first fossils of them in a formation earlier this year. So from the Cambrian, so that's pretty neat.
sure.
It's always
Yeah,
neat
but
finding.
that's what happens. You find some neat organism and you start studying it, and then you look up, and all of a sudden your beard is gray, and
Is that what happened with zebrafish?
that's what happened. Yep, yep. Just started plugging away at it, and before you know it, you're some old geezer.
What
So,
what's like the next group related to zebrafish?
zebrafish are cyprinids, so it's a fairly diverse group of little minnow-like fish all over the place. So,
Hmm.
what is the next group? I don't know what the next group would be. I don't know the exact phylogeny of zebrafish.
Oh, okay. I gotcha.
Yeah, and I don't know the precise phylogeny of spiders either, but that hasn't stopped me.
Oh, I don't know really. Uh, as far as like arthropods go, and I think for arachnids, there's like the or chelicerates, I should say. There's the which is the Pantetra pulmonata. I think that's one of the
Right.
hypotheses for their interrelationships.
Yeah. Yeah. At this point in my career, I'm just trying to figure out local species and how they're related to each other.
That's pretty cool. I mean
Yeah.
that's kind of the foundation of evolutionary theory. You know, it's
Yeah.
Darwin.
It's like we were mentioning earlier, working backwards.
Yeah,
Start at the species level and try and figure out how they're related, and then work your way back. And I'm very much at the species level right now. So like we have um, I work with four different species of spiders, and they're they all have these diverse origins. Most of them are not native to North America, but they just hitch rides with humans and go everywhere. And pretty soon you got this mishmash of these different populations. You know, it's all these immigrants coming in and wrecking things for everyone else, right?
careful! Don't say that too loud. Uh, yeah, that is really neat.
No,
Um,
actually, I love the immigrants because they're.
or do what?
What was
You said
that?
actually I
What?
love.
Oh, I actually love the immigrants because they're actually more interesting. The native species here, the one that's localized to just the northern part of the U.S. and Canada, is Steatota borealis,
Hmm.
and they're kind of cool animals, but they don't have the interesting pigment patterns that the others do. So, of the four species in the lab, they're the ones I'm least interested in studying right now.
I I've I guess I'll have to start thinking about in the pretty near future what because I'll be putting in applications to schools this spring. Yay!
Yeah.
And I really don't know what I have different ideas for things I would like to study, but I don't know. But it's kind of you know, it's a little daunting. It's like, hey, what would you like to do for the rest of your life? I know you're.
Well, I can just tell you how I did it, and the thing is, as a lowly undergraduate, you don't get to decide precisely what you will do when you go off to graduate school. So the thing to do is not worry about it, and you look for people who are doing interesting work now, and you apply to work in their lab, and they'll give you things to do, and you will gradually shape what you're going to do your research on, and you will be shaped by the place you end up in. So you know your destiny is not set yet.
Well, that's good. But
Yeah.
I do kind of like research into multicellularity. Um,
Uh-huh.
Maybe some of the lesser known multicellular critters, because oh okay, Nestling, what questions? Sorry, because I missed the chat, or I missed part of the chat because we had technical issues. Yeah, maybe some of the lesser known critters, because it seems like most of the research focuses on. Oh, is the Medusa stage of Nidarians ancestral, or was the ancestor like a sea anemone? Well, sea
Oh.
anemones are in, what is it, Octocoralia? They're in the Anthozoa group. They're either
Uh-huh.
Octocoralia or Hexacoralia. Oh, I think the I was saying I think the Arbilaterian was probably benthic. I was asking about the Armetazoan, if that one was planktonic. Oops. I don't know about the Medusa stage. That was an interesting paper by was it Kayal? It's like Kayal, 2018, where he did this whole phylogeny of Nidarians. Um,
Uh-huh.
it's kind of oh, but yeah, I think most of the focus is on like or most of the focus that is there seems to primarily be on either the plants and their algal relatives, the charophytes and the chlorophytes, and all those guys like Chlamydomonas. Um, but then on the other hand, you have the coenoflagellates, and it seems like every year they're finding new members of the other groups in the Opistocont lineage, new like
Mhm.
Philisterians and Ichthyosporians and all that, and they're just pushing back how far the cellular machinery for or that is used in multicellularity existed. So like we even share some stuff with amoebas, which is used in that. But there are other groups, and I can't remember any off the top of my head. It was like fungi, but I don't really like fungi a whole lot. Not a huge fan of fungi.
No, they're great on burgers.
I have to disagree there too. I'm sorry. I don't even like to eat them. I just have no
Oh,
culture. I'm sorry. Was it my botany professor said, "Oh, they're great and they taste great," and I was like, "Yeah."
well, wait. Yeast makes beer. How can you not like fungi?
Well, there's a thing. I don't drink either. You know what? It's like everywhere we go, I'm just making it worse. There's
Yeah. Uh,
there was one interesting paper on kind of the or on the origin of multicellularity in fungi, because they got the little chytrids who are actually
right.
polyphyletic now instead of monophyletic, but they're still got the cellular machinery which is being co-opted for building or for sticking um, cells together. But anyway, do you have any having like announcements or any things you want to add? Because we're at the hour mark.
Do I have announcements? No, I, I am planning to just spend the next couple of weeks playing with spiders.
That sounds
So
pretty fun.
it is.
I actually have a scorpion now, so yeah.
Oh, good for
We
you.
should get them to all play together.
Oh no, that's one of the tricky things about raising these things. You got to be careful about who you put in the same cage with who else, because they will kill each other.
They just keep biting each other's head off. Pun intended. Haha.
Yeah, it's one of the things that makes them awesome.
You know. All right. Well, I guess I'm gonna cut this. For anyone who came late or missed the first part, The Rocks Were There, the book that RJ and I are writing, won't be out for Christmas, unfortunately, but it will be out in January. So get a late Christmas present for your friends and family and fellow creationists, I guess. I don't know. Uh, well, thanks again for coming on.
If you're an atheist, you can celebrate Christmas any time of the year you want. So it all works.
You could, yeah. Holidays are just constructs, so
Yeah.
you can
Social constructs, you can defy them. Have
exactly.
Christmas every day.
I can't afford that after like maybe the first week, but
Right.
All right. Well,
Okay.
thank you again
Been
for coming
fun.
on. If you could actually stay for 1 minute after, but to everybody else, thanks, and we'll see you next time. Bye.